Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Great to be here again, everyone, on the Genetics Podcast. I'm really excited to be joined by Michelle Teng, who is the CEO and co-founder of Etcembly. They're a company that is working on immunotherapies by using machine learning to develop tailored T-cell receptor therapies. We're going to talk a little bit about her journey as a scientist and entrepreneur. She has a personal connection to the rare disease space as well that we're going to cover. So we've got a lot of interesting ground to cover today. Michelle, thank you for joining us. Looking forward to it.
Oh, it's a pleasure, Patrick. Thanks for having me on the podcast.
And my pleasure. I'd love to just start with the immunotherapy landscape more broadly. So this is something that people will be familiar with, I think, but probably not have a ton of detail. So I'd love if you could just talk a little bit about the revolutions in this field over the last decade or so, a little bit about how it works, and then maybe you could finish off by talking about where the gaps are, what work is still to be done, because there's still a lot of work to be done, and that's a big part of your focus right now.
Yeah, I mean, I can start from the beginning, right? So immunotherapy, I have to be honest, back in the 80s and 90s was just literally an idea. So it's taken a long time to get to a place where it is right now. But if you think about it, from a scientific standpoint, it makes a lot of sense because in the case of immunotherapy, it's all about breaking tolerance against cancer cells that have been seen as self for a very long time, right? It's basically being cloaked and hidden away from the immune system. So immunotherapies are great because it basically releases the brakes on the immune system and it allows the cancer cell to reveal itself and to be seen as a foreign entity in the body, right? So when you apply immunotherapies, the immune system kicks in and they start to recognize the cancer cells and start to destroy them. But also remember, because it took 20 years to get to a point where it's hugely successful and being like the standard first-line therapy in most solid tumors, it's very expensive, right? You're basically talking about investment to save a person's life, and thinking about the NHS, giving your bang for your buck, to make sure that it's going to give you the right outcome in terms of survival. It doesn't always give you what you need. So we know that it's often very toxic, first of all, and it doesn't work for everybody, right? So we know that we need to understand why there needs to be new treatments out there. So we need new biomarkers to work out whether cancer immunotherapy is working or not. But it also comes down to better patient selection. So you have to start with working out which patient set would work better with immunotherapies and others don't. So you don't waste money treating patients where it wouldn't work. So that means in the case where immunotherapies wouldn't work for these patient subsets, we need new targets. And ultimately, it comes down to a deeper comprehension of the underlying biology to identify new biomarkers and targets, right? To give better outcomes for more patients.
So what do we know? What do we know today about what percentage of people do respond and the reasons why some people respond or don't? Is there any window into understanding why? You can take the brakes off, as I like that analogy, in some cases, and the immune system takes over from there. But in other cases, it just doesn't work, or it doesn't work for long enough.
It's a whole myriad of reasons, right? You also have to understand what other pre-treatments or other, health history that person have. In the case where it doesn't work, it could be, tumor heterogeneity. It could be lots of other factors that is not that straightforward. Sometimes it could just be something as fundamental as it's just incompatible with that person's immune makeup or genetic signatures.
Right.
There's a whole variety of reasons, but ultimately, we need to understand why they haven't worked. And I think maybe it's not targeting the right part of the immune system. Releasing the brakes on the T cells doesn't always give you the push. Sometimes you need other factors, to push the immune system. Basically, it's basically there are lots of checkpoints in the immune system. You're releasing the first checkpoint, which is a key checkpoint, may help you release the dam, but it may just be a trickle, which isn't enough. The strength of that response isn't enough to eliminate the entire tumor.
Yeah.
So that there's lots of other reasons, right? And also tumor escape, for example. Once the tumor understands that it's being attacked, then it starts to evolve, right?
Right.
So it adopts new mutations in order to basically make tumor escape more successful.
Yeah. If you could just talk through kind of step by step the current generation of T cell receptor therapies, what's happening? So are they all, is everybody getting the same? Is everybody getting the same kind of reprogramming? Is there some difference depending on what they know about the individual's tumor and what's actually happening? I think people have a general sense of we're releasing the
Yeah.
brakes in the
Well,
immune
my God,
system, but
this is a whole labyrinth, right? I mean,
yeah,
it depends on the kind of tumor, but just generally, if it's hematological tumors, then there are like CAR T therapies, for example, that you can use as first port. But very often, I don't. I think the NHS will go for the cheapest option first, which is
yeah,
chemo, just to eliminate as much of the tumor cells as possible. And in the case of solid tumors, if it's resectable, they take it out first,
yeah,
right, with minimal risk. And obviously, resection presents another layer of complication because you can release tumor cells into the blood circulation and
right,
encourage metastasis. So they basically have to weigh out their options and see what is the lowest risk to give you the best outcome. And usually there is a chance that even with tumor reception that the cancer will come back in either in the same organ or in other parts. It will spread to other parts of the body. So the question is, sometimes there is a preventative option that comes post resection. So sometimes they give the patient time to recover, and then they give preventative treatment like a checkpoint in combination with cancer vaccines, for example. That's another area that's being trialed right now that could actually prevent relapse. But generally, most patients have to go through rounds of chemo before they come through immunotherapy, checkpoint inhibitors. Sometimes in rare cases where they feel like the tumor is not resectable, then they put in checkpoints as the first line. But checkpoints is becoming more and more the forefront of first line therapy with combination TCR-based therapies in the future, but also just in general T-cell engagers being another area that has received a lot of attention. And then obviously the other area that's been like the darling of immunotherapy or just cancer therapy at the moment is ADCs, where you
mm,
basically plant very specific nuclear bombs in certain areas and basically blow up the tumor cells, and hopefully that contains the spread of the tumor. That has received a lot of attention. It's currently what we consider, the forefront of cancer therapy. It's not quite immunotherapy. I wouldn't call it immunotherapy quite, but I think combining something like ADCs with immunotherapy could be another very effective way. It's basically coming at the tumor with different ammunition to make sure that it's
yeah.
contained and it's eliminated. A bit like warfare in a
Right.
way.
Yeah. Yeah. So an ADC in this case is antibody drug conjugate, right? For anyone who
Yeah.
wants to go down a rabbit hole and look that up. I'd love to talk about what you're doing at Etcembly. So you have a really interesting, I think, approach to making sense of all this, which is actually looking at long-term survivors of cancer. I wonder if you could talk a little bit about the origin of that idea. You run a study called Long-Term Survivor Study. How did that come about, and what's the purpose of that in terms of discovering new T cell receptor therapies or new ways to approach this space?
Yeah, I mean, if you think about the cancer survivor study, it makes a lot of sense because I've always wondered, and also the rest of the team have also shared the same thought line of thinking. Is if you kind of look at cancer survivors, they have basically been successfully treated with either immunotherapy or chemo, and they have kept the cancer under control, so it doesn't come back. And there's also been literature in the past where oncologists report that, if a cancer survivor donates an organ to a recipient, the recipient gets cancer. So you think about that, what could it be? Well, there's clearly some cancer cells lurking in their system that has been, transplanted into the recipient. So that's one area of thought that maybe, there's something unique about cancer survivors. And in the same line, if a cancer survivor develops a immune deficiency disease, then they also will see that the cancer comes back. So it means that there is something unique about the immune system that's keeping the cancer under control and preventing it from growing. And by extension of that thought, it's very likely that it's something in your T cell or B cell repertoire that's controlling it. I mean, obviously, your immune system is very complex, right? There could be other components, but very likely one of the key drivers of cancer survival is your immune repertoire. So we have, embarked on this large study of at least 100 donors, at least starting in the UK, but we want to broaden this to other geographical areas to really interrogate the immune repertoire in a way that's very deep. So we're sequencing millions and millions of T cells and B cells to uncover what could be the immune components, the clonotypes that are driving survival. And the way we're going to do that is through using machine learning. So we developed our unique machine learning algorithm we call Emily, stands for Assembly Machine Learning.
I like it.
Thanks. It's also named after quite a famous founder in the West Coast. I'll leave you to work out who that is. She knows who she is.
Emily.
Yeah. But she's basically, so machine itself can work out what could be the important clonotypes and also deorphanize them. It can stratify what we think are the common T cells and B cells that exist in all these individuals. So we're starting with HLA-A2 as a immune genetic background to start with, and then we will diversify them into the broader ones, not just the European backgrounds, but also the Asians and the Blacks and the Indians. And, so basically want to stratify the different ethnicities. I think that's where you start to uncover HLA-independent T cells and just antibodies in general, that give this protective immune signature. And
so how
send it to the drugs in the future.
Yep. How will you characterize, or how are you characterizing those? Is it single cell sequencing? Are you looking at, the kind of antibody repertoire that's presented? Talk a little bit about what you're expecting to uncover and how you're looking at these 100 survivors.
Yeah. So I think it's multi-step. It's not just, send training data into machine gives you answer. I think there's a lot of empirical validation that comes into play. So we do build like libraries of these T cells and B cells and try to deorphanize them experimentally alongside with machine learning. Anyone who thinks that you can just press a button and get the answer we're not there yet. We understand, we're realistic about what we can do at the very beginning while we start to deorphanize. So there is obviously the actual sequence space that you try to work out. At the same time, you're also doing a lot of autologous killing assays. So cross-tumor killing assays in this case.
Oh, interesting.
Basically express your T cell, TCR or BCR, and you look for tumor reactivity in a completely unrelated, a tumor from an unrelated individual.
Yeah.
It's this cross-tumor killing that will validate functional activity. So once we know that's functional activity, we sequence them as well alongside the agnostic sequencing to try to work out what it could be recognizing.
Yeah, interesting. So I think the best case scenario here is you find an individual or a group of individuals who basically has a naturally occurring antibody effectively that is able to kill a wide range of of tumor types, and so then you can then essentially use this as, like you mentioned before, an input to a drug discovery process to develop that antibody in a way that you could pull off the shelf, right, and treat people with. So the long-term survivors are kind of the discovery platform, and then based on what you learn, you may not take. I guess you wouldn't necessarily take the exact antibody um, into drug development, but it would be a great starting point for something
Yeah.
that could be
So that's the starting point. But we
yeah,
can also engineer,
yeah,
immune receptors using machine learning very well, very quickly as well in a matter of months. So it gets turned into, either bispecific or, we choose a
yeah.
modality that we think it's suitable for, and then we start creating new therapeutic modalities from there.
Yeah, you talked a little bit about essentially this is kind of an analogous problem to the large language models that we're used to, at least I'm used to interacting with every day now. It's become a big replacement for my Google search. Talk a little bit about that analogy. Like how do you use large language models? What is we all understand that ChatGPT reads all of the internet and then can finish sentences. What's the analog to that when it comes
Yeah, I
to
mean, if
understanding the
you're
immune
thinking
system?
about large language models, it makes a lot of sense for immune repertoires, right? Because it's the language of TCRs and BCRs and how it
Yeah.
recognizes. It's basically sequence matching sequence. And in the world of large language models, the language of amino acids can be learned very quickly. So we're doing basically protein language models here and matching up sequence space between the TCR BCRs with its target antigens. In terms of like what it can do, we start with building repertoire networks in terms of relatedness between clonotypes. And
Hmm.
once you have these networks, we can also infer specificity because we have known specificities for certain clonotypes that are, well known, well characterized, and also characterized by our cells. And you start to infer specificity from degrees of separation from the core central nodules that have known reactivities or specificities associated with these clones. And as these networks mature and you start to build new training data set with known specificities, you start to fill in the blanks between, slightly related clones to the point where they're completely unrelated and you know what they are seeing, recognizing in the immune system. So it's a stepwise gradual process. You don't, you won't immediately deorphanize the immune system. I think we're still.
When you say, sorry, I'm not familiar with the term deorphanize. Can you define that for me and for anybody else who's listening who's like me?
Yeah, so I think when we talk about deorphanization here, we're basically saying what is a T cell or B cell receptor recognizing? What is
Yeah.
it seeing in the immune system? Right, it's basically like a puzzle, a bit like the Enigma machine, what's the
Explain.
code? So we always call what we're doing code to cure,
Yeah.
from deciphering code to finding a cure.
So basically, when you like, if you were to sequence my T cell repertoire, you'd find a lot of things that you're basically not sure what its purpose is, what it actually targets, and it's that process of understanding like what is it targeting? Am I getting that right?
Yeah,
Yeah. Okay.
exactly. But also, quite interestingly, this is not really spoken about, is that everyone, regardless of our genetic background, have about 10 to 20% of our T cell repertoire to be public. So everyone has
Oh,
them, regardless
interesting.
of your background.
Yeah, I was going to ask about that. Okay, so 10 to 20%
Yeah,
is shared. If you sequence
share.
mine and yours, we'd have them similar. The other 80 to 90% though, is that totally unique to us, or what are the
Yeah, we
how does
call
it
them
cluster?
private clones. So
Yeah.
basically, they are probably HLA restricted ones, but the public ones are, probably seeing T cell clones, T cell clones that recognize antigens that are presented maybe in an HLA independent manner, for example.
Right.
Those are extremely interesting because they are probably also driving immunity in some way that we don't quite understand yet. Or there is some, literature out there that suggests the HLA independent TCRs can actually mount a meaningful immune response in the case of cancer, or even you can turn it into any other kind of drug. So that's again very poorly understood anecdotal evidence from publications in the past that needs to be further teased apart.
Yeah. Can we talk about the HLA for a little bit? Because as a human geneticist, it's a very scary part of the genome to me. But you did your PhD in immunogenetics. Can you
Yeah.
talk a little bit about there's obviously we all have there are different HLA haplotypes. They're incredibly complex, can be difficult to sequence, but they have a really important role in almost every immune related disease that we have. Can you talk a little bit about that complexity and how it relates to to differences in T cell receptor biology, because it's a big gap for me. And I know there's a connection there, but I don't know it very well.
How much time have you got?
We got 5 or 10 minutes. Give us the
Okay, I'll give you
immunogenetics
a
PhD
very
101.
okay, all right. I'll give you a very generic overview. So our HLA background is, well, in the human, it's a
Yeah,
human leukocyte antigen, right? It presents a variety of peptides that is produced by your cells. So your every single cell in your body has this turnover machinery. It's like your recycling center, right?
yeah,
So it recycles proteins that have been worn out, or it's basically has expired, and it churns out from whole proteins into small little peptides, and these peptides get loaded onto HLA molecules, right? And we have very diverse HLA molecules too. In a way, it's a protection, because in order to present your HLA molecules peptides on your HLA, it needs to go up to the cell surface and tell the immune system that everything is fine.
right.
Now, in the case where you have, say, a viral infection, in something that everyone understands, like for example COVID, if you have COVID, you will start, your immune system or your cells will start presenting COVID peptides,
Yep.
and that's when your T cells and B cells get alerted. Okay, foreign antigen here, foreign body. Let's try to eliminate this cell that is infected with COVID virus, and that's where your immune system can actually come in and mount the right outcome, the right immune response. Now, the other aspect of T cell diversity goes through thymic selection, and my God, we can spend a whole 45 minutes just talking about thymic selection, right? So all of us are born with diversity of, say, 10 to the 8 T cells. That's well known, but that repertoire shrinks as we grow older. Right. So we start with the 10 to the 8, it goes to thymic selection, where thymic selection actually selects for the best T cell clones that are not self-reactive.
Right.
And that's important, because you don't want your T cells to be mounting an autoimmune response. And over time, as you grow up, that immune system matures, because it gets exposed to, viral attacks, environmental antigens, as you grow older. But when the T cells mount an immune response, they expand, or they become memory T cells, which is what's protective in most cases.
Yep.
But very often, as you grow older, these immune systems, when they mount an immune response, they don't actually restore itself. So some of these memory T cell clones die, which is why your T cell repertoire shrinks as you grow older. Also, anecdotally, interestingly, there was a study with centenarians in Okinawa, and they
Yeah.
showed that the immune repertoires of these centenarians actually have a very diverse T cell repertoire. So obviously, it's the diversity of your T cells
Yeah.
are protective and give you a form of immunity that gives you, ability to kind of live healthily for a long period of time. So we know that the immune system is extremely important in, general health.
Yes.
And keeping
This is super helpful. Thank you. In the long term, coming back to the long term survivor study, what do you know about the T cell repertoires in that population? Are they more diverse, less diverse? I know they probably go through, like, they've gone through chemo or some other things that could hugely disrupt that population. So I realize what you learn is not just a factor of they've survived the cancer, but also many other things. But I'm curious of what it looks like if in that kind of population.
Yeah, so that's another interesting set of studies where, patients who have had TIL therapy, so tumor infiltrating lymphocytes, as they're called, when certain sorts of patients receive TIL therapy, we find that TIL therapy works very well when these TILs are actually very diverse compared
Interesting.
to, TIL repertoires, which are very much shrunken and, sort of clonotype driven. So if they're very dominant clonotypes, the likelihood of mounting an effective immune response is a lot lower, which also kind of points to the fact that the older a person gets, immunotherapy may not work as well, because your immune repertoire isn't as diverse. So you think about different types of artillery in your immune system. They basically have shrunk from what it was, in your 20s and 30s to when you are in your 70s and 80s. So that's why immunotherapy may not work as well in older people. And it's so important that having a diverse immune repertoire means that you can actually mount effective immune responses against most kinds of antigens that you encounter throughout your life, whether it's cancer or virus and beyond.
Yeah, interesting. So coming back again to the LLM, I would love to just understand a little bit more about the large language model. Is it, literally as my simple brain is imagining, there's a chat GPT like terminal where you're saying this is the population that, or the type of tumor, for example, that I want to target. Please generate candidate sequences for us to go test in the lab? Is it embedded in some kind of longer stepwise process where you're generating and then you're testing in the lab and then you're feeding the results back and regenerating new hypotheses? I'd love to just learn a little bit more about how that
Yeah,
works.
so I think the large language model in itself is just one of the many different
Yep.
steps that we embark on, right? So first of all, you have sequence based, but also you also have very sophisticated docking models in our case, where we can in silico model antibodies and TCRs interacting with its target and
Hmm.
measuring affinity and likelihood of interaction. So once you've got that stratification and that table, you can test them empirically in the lab, and the results and outcomes from testing gets fed back into our machine, and
Yep.
we refine the models that way. And it's very important because when you're looking at large language models, even you look at ChatGPT as an example, right? If you ask ChatGPT a question that is available in the public domain, they can give you a very reasonable answer. However, if you're asking ChatGPT to find you immunotherapy options for X, Y, and Z kind of tumor disease, you're going to be struggling because
Yeah.
they don't have that sort of specialist knowledge, and that's what we're building. We're building a large language model with specialist knowledge in immune repertoire sequences, and also training it to understand what specificity looks like.
Yeah, it's
That's
amazing. And you've got
nuance.
a nice feedback loop as well, right? Where you've got because one of the challenges with these models in biology is we don't have an internet's worth of text data like you have for ChatGPT. But if you have a feedback loop where you can test in the lab, feed it back into the training model, generate again, test in the lab, feed it back, then you do have an interesting way to generate this really specialist data, over time, iteratively by actually doing the experiments, right?
Yes, that's right. And that's where, what we're doing is really unique because we're also building a chat GPT model. So we have an interface
Yeah.
what where we can interact with our large language model and ask Emily to find TCRs against a specific antigen. And Emily can pull them out, whether it's in the public domain or within our database. And then we can ask Emily to run pipelines. So she can pull in tools from the open source, like MPNN, for example, and ask Emily to find the most likely mutations that will enhance specificity. And so Emily can churn out these answers very quickly in a short space of time, literally in like 20 seconds. So she can come back with a list of answers and you can test them up empirically. And you can also ask Emily to pull in tools like, can you send these answers to Slack, for example, or send it to Jira
Yeah.
so that we can, set up a new experimental workflow after, creating these predictions. So basically you automate discovery, engineering, and also testing, up to a certain point on the system. And there's just human intervention in validating and doing the experiments. I mean, you still need, human input at the end of the day.
Yeah.
Training data at the end, is the most important component that defines success of predictions, right?
Yeah, that's right. And you prior to founding Etcembly, you were at Immunocore for a long time, right? Which is one of the pioneers in this
Yeah.
space. It'd be great to hear a little bit about you were group leader there. I think what did you do there, and how are you taking that, I guess generation one that you worked on there and bringing it forward. I think they're investors and they're on their board, so on your board, so they clearly believe in what you're doing and think there's something interesting here. But that's quite a big company, right? What has it been like to go from the big, relatively big company in a biotech world to now starting and running your own company? How's your day changed?
Well, wow. Well, where shall we start? I've been in pharma for quite a long time, overall in the whole industry. So I started my career in AstraZeneca. Back then it was called MedImmune, Cambridge Antibody Technology. So my role there was to basically, carry out phage display for discovery of antibodies at that point. And I worked on FluMist for quite a number of years there before I moved to Immunocore because I wanted to experience how small biotech operates. And it also had this amazing culture and atmosphere that drew me to the place. So when I joined Immunocore, it was actually our CSO, Nick Pumphrey, who interviewed me. And I love the story because I remember him very, very distinctly. And when we started the interview, when I was interviewed by him and Ben Jacobson, the founder of Immunocore and Adaptimmune, it was quite a profound experience. And I was really struck by how passionate they were about the journey, the experience of, putting TCR therapy on the map of cancer therapy. And I eventually joined, a couple of months later, and my role there was again to basically do phage display for TCRs. And over time, it was apparent that the way TCRs are found and discovered is extremely laborious.
Yeah.
T-cell cloning is kind of like the main port of call. You basically do like 9 months of heavy experimentation. You may be able to troll out 1 or 2 TCRs, or maybe sometimes when they're successful, you have like more than a handful, which is a good run.
Yeah.
But its success is never guaranteed. And then once you do some testing in the lab and you're relatively confident that it's specific, then you go through a phage display to affinity mature the TCR. And that takes another 9 to 12 months, depending on how well the TCR behaves in the context of a phage library. Again, painful experience. You get to a point where you have lots of variants, there's lots of testing, and it's kind of like high effort, but reward ratio is kind of like, is high in effort, but reward is never guaranteed, right?
Right.
And I thought, well, there's got to be a better way to do this. And so around that time, single cell sequencing technologies were coming to the fore. And so I decided to let's say, why don't we try single cell sequencing as a alternate to, finding T cell clones? And you can do it more rapidly, and you basically eliminate a lot of the heterogeneity that comes from clonal T cell testing. So that was kind of applied and deployed in Immunocore as the standard way to find TCRs in the longer term. That's where I built the team around. But as I worked in T-cell cloning and TCR discovery for, the next couple of years, it was very apparent that there's certain kind of chain pairings that were converged towards certain antigens. And so the hypothesis was formed around, well, maybe there is something here you can apply mathematical models around. And I kind of, brought this idea to Jake Hurst, our CTO co-founder. He, at that point, was working with me on various projects, particularly around Kimtrack. We were trying to work out the mode of action of that molecule that was still progressing through phase 2 trials at that point. And we both agreed that there's probably something that can be done using machine learning here. So the idea was born, for in Immunocore's case, when we proposed it to them, I think there was, there was less appetite to take on something so novel, right? So they were becoming a clinical stage biotech, and I totally understand that it's probably not the right time for them to explore it in so much detail. So, and around that time, I was about to go on maternity leave. So I decided, well, I thought about it to a great extent when I was on maternity leave. Like, what should I do? Should I go and find out for myself? And Jake also left Immunocore at that point to take a career break. So the timing was absolutely perfect. When I emerged from maternity leave, I thought, well, I can find out or I can go back to where I was. And I took the plunge and decided, well, life is too short to wander. So let's try to settle a company and see how far we go with this.
Yeah. And that was January 2020, right? Which was perfect time to start something new, because a month later, the whole world shut down. What was that like?
Yeah, I remember large language models didn't really exist in the current flavor,
not at all.
And also the whole idea of AlphaFold was just an idea, academic idea at that
Yeah.
point. So I think we've come to the right place at the right time at this
Yeah.
point. The intersection of large language models, AlphaFold, and structural modeling being so sophisticated and advanced right now. We are able to even visualize our molecules using VR, for example,
Wow.
doing real-time manipulation coming off Emily, and just basically playing around with it literally with your hands, through
Amazing.
VR goggles, has also been quite an experience.
Yeah. So do you use AlphaFold to generate structures when you're looking at like the kind of docking, the kind of docking mechanism that you spoke about earlier, or is that, how does that fit into things?
Yeah, so AlphaFold, to their credit, being open source, has
Yeah.
actually, helped elevate everybody into the space where everyone has a very sophisticated model. I mean, AlphaFold 1 and 2 didn't perform as well as our own in silico docking models. So we started to develop our own models that were more amenable for TCR antibody docking. I think AlphaFold 3 has come on quite a bit,
Hmm.
and probably would match what we do. But at the moment, we basically trust our own models because we developed
Yeah.
from scratch, and we made a lot of crystal structures ourselves to basically fine tune the models. So in a way, you need foundational models at AlphaFold to be out there for people to learn from, and then
Yeah.
they can fine tune it for their own purpose.
Wow, it's amazing. It's a very cool kind of chain of models and techniques that you have together. It seems like you can do, like you're taking things that 10 years ago would have been an entire PhD project, right? Let's figure the docking out of this single thing, and now you can run in silico predictions, test it in a much more high throughput way. It's exciting. I mean, it means you can generate hypotheses at a rate that's order of magnitudes higher than
Yeah,
before, right?
absolutely. But interestingly, now that you've overcome the bottleneck in discovery, the bottleneck moves further down because we can discover,
Yeah.
engineer immune receptors so well. The bottleneck goes down to the preclinical, and then that becomes the next area that the field has to overcome. I still think, that takes its time because experimental validation to produce, a preclinical package for the FDA still needs the input of effort, labor, time, and resource.
Right. What is the bottleneck specifically there now? Like which part of the preclinical package is the hardest?
So I think in most cases, you need to kind of demonstrate, specificity, and there's no cross-reactivity. So you do a lot of preclinical work in a very extensive panel of cell lines or primary cells to show that it's only recognizing, the antigen presented on um, antigen positive cells, right? And not in healthy cells. That's one area. But nowadays, I think most companies are doing some degree of non-GLP toxicity studies. So doing some baseline studies in animal models. Or we try to steer clear of that because we don't believe that animal models is the way forward. I know that the regulatory bodies do require single species toxicity in most cases. But to Immunocore and Adaptimmune's credit, they've managed to argue that out of the preclinical work with the
Oh,
FDA,
interesting.
and they have accepted it, So we still abide by that sort of thinking to minimize or completely eliminate animal testing if possible for this sort of work because it's not necessary and it's also not biologically relevant. Yes, you great, you killed a mouse, but let's kill the humans, right?
Right.
So your most compelling study is your first in human, not your first in mouse or
Yes, that's
something
right.
like that.
Before we wrap up here, I want to also talk about a second company that you're involved in. You're staying very busy. So in 2021, you founded and you're the chief scientific officer of SynaptixBio. You've shared before. I think that this is in part driven by the fact that your oldest daughter has a form of leukodystrophy that's life limited, and you done some work to try to raise funds charitably to work on treatments for this condition. But I think you figured out that a company was probably the best long-term, most sustainable way to actually try to really solve this problem. So I'd love to hear if you're happy to share a little bit about the story there and what drove you to start the company.
Yeah, I'm happy to share that. So Sophia was born with a mutation in a gene called TUP4A. It's basically a tubulin cytoskeleton gene that is expressed in the brain, primarily around white matter. So when we say white matter, it's the myelin that coats the nerve cells, right? And the myelin itself isn't forming properly because of the mutation of this tubulin TUP4A gene that transports myelin to insulate the sheaths. It was a very long journey, to be honest. When she was born in 2010, she kind of had a very normal childhood until the point where around the age of two and a half, she wasn't walking stably. And we decided to get it checked out by the pediatrician. And the doctors couldn't really work out what it was. And it was through a combination of eventually MRI scan and genetic testing that we found out what it was. That was a spontaneous mutation that wasn't inherited by myself or my husband. That occurred during development, and which led to what they call a toxic gain of function. So she only has one copy of this gene. The other copy was normal, and that led to eventual diagnosis, which incidentally was I diagnosed her, not the pediatrician, because at that point, the papers around typhoid leukodystrophy, or HABC as they call it, is not even, it literally just came out in 2014. Wow. And we sequenced it around that time. So I trawled through literature to find out what it was, and eventually landed on that paper that was published by Marjo van der Nap in Amsterdam, along with one of her protégés, Adeline van der Ver, who we're still working with at the Children's Hospital in Philadelphia. When I found that paper, there was sort of like this moment of relief and enlightenment that you finally understand what you're dealing with.
Yeah.
And I remember printing that paper out and then bringing it to the clinic with Sophia, and I showed it to the pediatrician. I said, this is what she's got. And she said, well, you're right. Oh, well done on finding out. And I thought, well, why does it have to be the parent? But, this, I guess, to no fault of anybody, it's kind of a reflection of how much education even like neuropediatricians need, because they are not always on top. They are constantly seeing patients, right? There's no way that everyone knows everything at that time. So once we had that diagnosis, I was in shock for quite a number of years, and I couldn't really bring myself to talk about it. And I didn't want to tell anybody at work either, because, I was worried about what repercussions it had for me. But eventually, I came to a thinking where I had to do something about it. So I started the HABC Foundation along with another patient parent, Amy Sheridan, and she's incredible. She's been, kind of been my emotional rock for such a long time. When we started the foundation, we wanted to raise money for research, right? And when we started out, we managed to raise we started the foundation just before COVID, and we managed to raise something like £200,000 over a space of 12, 18 months, which was incredible given that everyone's in lockdown. And we donated quite a lot of that to CHOP, Children's Hospital, Philadelphia. And they were doing quite a bit of research around the natural history study, and some, also understanding mouse models that mimic the typhoid leukodystrophy condition. But it became apparent that there was no way we can actually bring a drug to clinic using the drips and drabs of drip feeding, small sums of money. Yeah. Even though it's six figures, it's still substantial. There was no way we could bring a therapy to clinic. So I, along with a friend of mine who's also a board member of Etcembly, Ben Jacobson, he basically said, well, why don't we just try starting a company to bring this therapy into the fold? And so Synaptix was formed, in 2021, and we managed to raise something in the order of 10 million. And we've got a lead candidate now that we're hoping to start trials with, in late 25, early 26. But it's been an arduous journey. I put my CSO hat on.
Yeah.
And, in the capacity of Synaptix, and that saves me, because you feel like you had the right calling in life. you wanted to be a scientist and this is what you needed to do. So that brings a lot of fulfillment and purpose in
Yeah,
your
it's
work.
amazing. Amazing that that all the progress you've made as well. You've got a candidate. It's an ultra rare disease, right?
Yeah.
I'm reading from based on my background reading. There's probably less than 100 individuals that have been documented in medical literature, right? So it's very rare.
Yeah. But interestingly, since the publications have emerged, we're starting to see some young adult patients now. So
Yeah,
patients in their 20s and 30s that have come forward or have emerged, from speaking to our charity or the charity in the US. So the numbers are actually far higher than we
good.
anticipated.
Yeah. Yeah, that seems to be a theme in a lot of diseases like this where actually, especially if the presentation is variable, then, there could be a lot more people affected than you realize. Have you started to look in population databases, UK Biobank and those sorts of things yet to understand just how many people might be out there?
Yeah, I think that's something we're starting to look at, but because, top 48, well, sequencing of the genome has not been that common so
Yeah,
far. And very often the D249N mutation that we're talking about here, by the way, there are lots of other mutations in the top 48 gene that can result in leukodystrophy too in the same gene. We've all literally, this is kind of like the icing on the cake. We're just literally just scraping the surface. So once we start looking into biobanks, which we're going to start doing very soon, I'm sure we will start to identify far more patients than we originally thought there would be. It's the case with uveal melanoma as well. Just a separate example where I worked on Kimtrack. They thought that uveal melanoma was a rare cancer, but
right.
actually, once the cancer therapy was out there, the patients have just come in droves. So it's very often that when there is a disease therapeutic treatment for something that's perceivably rare, that the patient numbers actually go up because people hear about it and they come forward, especially with a condition that's so new.
Yes.
So, it's only been described, literally about 10 years ago now.
Wow. And yeah, where there's no good treatment. Just to connect the dots for everyone, Kimtrack, but correct me if I'm wrong, was developed by Immunocore. You
Yeah.
were highly involved in this, so it's a T cell therapy for a subtype of melanoma, right? Uveal melanoma,
Yeah, it's a TCR-based bispecific for uveal melanoma.
right? Just to wrap up on Synaptix, I was curious. You mentioned that you have a lead candidate. Are you able to say, is it a small molecule ASO? I'm just curious about what, because you've got different options when you've got a gain of function like this, right?
That's right. So we had lots of different options when we started Synaptix, and AAV was one of them. That is kind of like a one and done, right?
Yeah.
It sounds attractive, but really there's still quite a lot of tox issues associated with AAV therapies. I think the new generation of AAVs look far more promising. Safety, efficacy, they basically tick all the boxes. But at the time when we started Synaptix, I think the only data points we had were, from very few cases. And one of the outstanding studies was on Millicent. I don't know if you know the story about Mila.
No.
She basically went from diagnosis to treatment in under a year by Tim Yu at Boston Children's
Oh,
Hospital.
I do know this story. Yeah, and they started a nonprofit, right?
Yes,
Yeah.
yeah. So Julia, the Mila's mother, um, started Everyone Medicines, which is a foundation, and I kind of, half foundation, half company set up to kind of help bring rare disease treatments into reality. I mean, sadly, Mila passed away a few years after the first treatment was done, but it's because the treatment got to her very late in the point where the regression was kind of happening in a very rapid pace. But for me, to just see Julia just carrying on the legacy to just save other children, it's such a moving experience to just be, to be part of her life. We still keep in touch regularly. She's kind of like another pillar of support in my journey. And she's also given hope to lots of other parents, just to say, keep fighting. And you need to basically fight for your child, because if your child has a rare disease, pharma industries don't really care about, doing a therapeutic for, a handful of children that are affected by the condition. But that's where patient-parent groups are so strong in numbers, and if they can band together and work together to find a treatment and a cure, and that's when it will bring rare disease into the forefront of genetic medicines. So yeah, we did pick an ASO in the end, and I think that was basically because practical reasons that it's kind of past the regulatory approval path, and it's well trodden and it's well understood by the FDA and other regulatory bodies.
Amazing. I can't agree more on the theme and the sentiment that you put across about patient organizations and how important they are in driving the science forward. There's a great episode that I reference all the time that we had. It was episode 105 with Warren Huff, who is the CEO of Reata Pharmaceuticals. They developed a Friedreich's ataxia drug that was ultimately approved, and it was only, and he says this very clearly in the episode, it was really only possible because the FARA, the patient organization, ran an incredible natural history study for years that built the evidence base that helped them to take it through trials. So it's so important that groups like yourselves start early, galvanize parents and donors, and get started on the science that often industry won't fund, because then you've really just you've laid the foundation and raised the opportunity, right, for somebody to go in and actually make something out of it.
Yeah, that's right. That's so important. You have to fight the corner for your children.
Yeah, I couldn't agree more. Michelle, thank you. This was an amazing episode. I learned a lot in two very different fields. So well done to you as well for wearing both these hats at the same time and making it all work. I don't know how you do it, but please keep doing it. The cancer patients and children like your daughter with rare diseases are depending on it. So thank you for you dedicating your life to these important pieces of work.
Oh, thank you, Patrick, for having me on your podcast. It's a pleasure to share the story.
It's my pleasure. I'd love to have you on in the future when you start to generate data from either of these and talk about how it's all going.
Oh yeah, I look forward to that.
Great.
Not long from now.
Thanks, everyone. You can find all the links to Michelle's work in the show notes. And thank yous, as always, for listening. We'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.